Okay, so hopefully now you can, okay, good. So just a brief outline. I'm going to give a very brief introduction to the 4D nucleome as a consortium and as a scientific effort. I'm going to talk about how you might find data of interest to you in the portal. Talk a little bit about the metadata we have and how you can visualize the data on the portal. How you can actually download data if you do want to use it. A little bit about our standardized bioinformatics pipelines and processing, and then I'm going to wrap up with how you might explore data actually in the cloud via our portal using our Fordan Jupyter Hub. So briefly, this is the program snapshot for the Fordan Consortium. It's a common fund program. And it's working to understand the principles behind the three-dimensional organization or architecture of the chromatin in the nucleus and how that changes over time, and especially developmental time or progression into disease and questions like that. So here's a cartoon of, kind of how we think about this in some sense. So there's a nucleus. It has chromatin, chromosomes organized into territories or regions, and some interesting structures like nuclear bodies and the nucleolus and the nuclear lamina. And then if you zoom in a bit and you start to look at the chromatin, you can broadly categorize it into compartments, the A compartment being characterized by mostly active chromatin, where you're getting gene expression, and the B compartment, which is inactive regions, also known as heterochromatin primarily. And then further zooming in, you get to the level of regulatory loops where you have your gene and enhancer interactions, and these can change to change the regulation of gene expression. So, and all the research that's being done by the 4D Nucleome Consortium members, researchers, are aimed at getting at these various levels, and there are different techniques and technologies for looking at these different levels. So phase one of the consortium ended almost 2 years ago. That phase of the project was largely driven by technique development efforts, as well as generating data, focusing on a subset of commonly used cell lines, so benchmarks and such could be developed. And recommendations about the assays could be developed, be provided. Now, phase two of the project is just starting a second year, and that has more of a focus on answering biological questions having to do with 3D architecture and changes therein as it pertains to human health and disease. To do this, we have a lot of data in our portal from two basic or broad modalities: sequencing-based assays and imaging assays. The bulk of the data currently in the portal is of the type of sequencing, and it was generated during phase one, although we do have some imaging data, as you'll see. The sequencing technologies are focused on chromatin conformation capture, or high-C assays, which are the workhorse of elucidating the three-dimensional structure of the chromatin. And so on the top left here, there are various variations of this high-C technology, data sets that we have in the portal. And on the right, there are also ways, techniques to actually enrich various regions where you're looking at the chromatin interactions in a subset of the genome, either those regions associated with particular proteins, targeted proteins, or in actually specifically targeted regions of the genome using Capture-Hi-C. There are also been developed some ligation-free techniques like GAM and Sprite that look at these chromatin contacts. And they're particularly useful for looking at multi-way contacts, so not just pairwise. And then other technologies like MARGI look at interactions between different molecules, RNA in MARGI's case, and the chromatin. All these experimental assays and techniques can be, the results are shown over here on the left side. You get these matrices of data that can be represented as these contact maps, where you're essentially showing genome versus the genome, and along the diagonal, you have high contact probability. And as you move away, the contact probability generally decreases, but you do see regions shaded darker that are locations of higher contact probability. To complement these chromosomal capture assay, Hi-C type assays, we have additional genomic assays available in the portal. Those that look at the position of chromatin relative to nuclear structures like the nucleolus or the lamina. So the MID and TSA-seq being examples of those. Those that look at replication timing. Those that look at protein factor binding sites, so cut and run and ChIP-seq, and chromatin accessibility and TAC-seq, and gene expression RNA-seq. And those are more typically viewed in a browser as one-dimensional tracks, as shown below. That's the sequencing data. We also, especially moving into phase 2, we're getting a lot more imaging data into the portal because the experiments are generating, the researchers are generating that type of data. So particularly in phase 2, especially of the project, there seems to be a lot of people doing multiplex FISH or so-called chromatin tracing type experiments. And I've just listed the variations on the techniques that are used for this chromatin tracing data. So we're continuing to get a bunch of this data, but we also have additional imaging assays in the portal, like DNA or RNA FISH, immunofluorescence, single particle tracking, and so on and so forth. So with that brief introduction, I'm going to attempt a live portal demo. You know how these live demos can sometimes go, but we'll hope for the best. So this is our homepage of the Fordan DCIC data portal. Okay, I'm not going to focus a lot about aspects of this page because we're currently implementing a redesign of the page. So a lot of things will change, and our aim is to help it make it easier to find and access the data sets that you're interested in. But some things that won't change are going to be the toolbar, where this toolbar is available on every page. So you can see the search bar and there's tabs that you can look at, and I'll be referring to these through my demo. Also, just a general count of the data available. So we have over 1,900 publicly available experiment sets, comprising over 4,600 replicate experiments with almost 37,000 files associated. The other thing that won't be changing is this bar graph. And so it gives you kind of a snapshot of the data available. So you can see in situ Hi-C data is our most popular data type, and that's not surprising. But we also have several other data types. Now, as we get into looking at how you can find data, I want to point out that this bar graph itself can be very useful. I encourage you to play with the bar graph. You can change the grouping categories. You can change both the X and Y axes. So you can kind of refine what you're looking at overall. But for example, say you were interested in specifically TSA-seq data sets. If you hover over the bar graph and then select it, and then choose the browse button, you'll actually be brought to a result page for all the TSA-seq data sets in the portal. So this is one way you can start getting at the, getting at the data that you're interested in. The other thing I wanted to point out here is the search bar. Okay, so when you click on the search bar, this interface comes up. And so, for example, maybe I'm interested in looking at data from hamster. We have a paper that was recently published by the Lombardis lab where they did experiments that's related to COVID, and there's some hamster data. And you can choose this from different types of items that in the portal that you might want to look at. So I want to look at experiment sets from hamster. If I do the search, I see results, but hey, wait a minute, I have both human and hamster data sets. The reason for that is because this is essentially a simple text search, and in these human data sets, which are also likely from the Lombardis lab, they were from the paper that described the hamster data as well. So a simple search brought up those data sets. However, you can easily click on this guy and refine and get only the hamster data sets if that's what you're interested in. However, our recommended way for actually getting at the data is to use our data browser, browsing all data. And when you do that, you're brought to a page of results of all our data sets. So we have 1,922 data sets. You see this table, which has some relevant information about the data in columns here. But I'd like to focus on the properties over here, which are essentially supports our faceted browsing features to help you narrow down and filter what data sets you're interested in. So you can filter by lab. So if you wanted to see all the data sets from Job Decker's lab, you could select that. Experiment type. So it's, very similar to like shopping on Amazon or what have you, faceted browsing. But I also encourage you to look at some of the other maybe less obvious sort of facets. So we have sample type. Say we're interested in just experiments done on tissues isolated from an organism. And maybe we're only interested in brain, tissues from brain. So you would use the organ system facet and select brain. And you can see that we have 17 data sets in the portal of various assay types that are from brain tissue, derived from brain tissue. So this is a pretty powerful way of filtering down and finding the data that you're interested in. We've also generated some specific pages that gather particular types of data together. So in the resources section in the data collections, we have some pages that we created. One where all the Hi-C type data sets are listed that are in the data portal, one for high resolution data sets. Let's look at the Hi-C data sets. So we have them categorized by single time point and condition, time course, disrupted or atypical cells, and those that are high resolution data sets. These we thought might be particularly interesting, so they get a page of their own. In fact, there goes with over 1 billion reads in the experiment. Another collection page I wanted to focus on was the chromatin tracing data sets. Okay, during phase 2, there are a lot of researchers, several labs doing this type of experiment, multiplex FISH experiments. So we currently are building our collection of this data, but we currently have 17 data sets or experiment sets of this data type. The interesting thing or an interesting thing about this particular data is that the data associated with it is process-derived files that are all in the same format. That is this Fordan FISH omics format that has been developed by the DCIC in collaboration with the imaging working group, as well as the labs that are actually producing this data type. And this provides a standard way to represent this data so it can be reused by reused by other people, and tools can be developed to use it. So I encourage you to check that out and read about this format if you're looking at these data sets. I would not be surprised if one of our later tutorial series actually just talked about tools that are using this data potentially in conjunction with genomics data to look at some integrated questions about the architecture of the gene. All right. So I've went over several different ways that you can try to find data of interest to you. Okay. Now let's look at this is a particular page for a particular data set. It's one I like to use for demo purposes. It's in situ high C performed on human embryonic cell line, stem cell line H1. This is the so-called high C 2.5 protocol on this. You'll see if there's a publication in which this data set was reported, it's indicated. For many of our assays on the pages for the particular data set, we have a little description of the assay so you can learn about how in situ high C works if you're not familiar with it. Some basic information, but I really want to focus on this tabbed region of the data set. So the default open region for any data set that has standardized processing on it done by the DCIC, the default state is to open to the files generated by that processing. So the process files tab. Okay, so you can see a list of the process files associated with this data set, either with the set as a whole or with the individual replicates. But I initially want to focus, well, let me point out that when you see this icon here, that's indicated that the file is visualizable in a browser, okay? For most of them, they're visualizable with our high-class browser, and I'll talk in a second about how you can look at them over here. But I did want to point out for every high C data set, we also produced a high C type format file. And if you really are, prefer to use like the Juicebox browser provided by the Aiden Lab or the FA Genome browser from WashU, these are link outs to view this file in those browsers. However, we at the Fordan DCIC really like high-class. You can see a view right here on this section of the page, and it's interactive. You can zoom in, zoom out. However, it gets a little cramped. So using this Explore Data button, okay, you actually go to our visualization space here, where you get a much larger view of the that same data. Okay, and so you can zoom in and zoom out and scroll around, and you can see the other data tracks. There's compartments and boundaries shown here, and most relevant or useful potentially is you can actually add data using any data file that's on the portal. So when you click the Add Data button, this window pops up, and then I'm going to use the facet for the file properties, look at the file formats, select mcool files, and I just happen to have previously looked at this, so we might want to look at a mcool file from a tethered chromatin capture experiment, along with our Hive-C. And if we do that, now we have both panels in view. So we're looking at data sets side by side, and we can scroll around and they're locked together. So that's a very useful feature. One thing I did want to point out are the save and clone buttons. They're currently grayed out, and this is one of the few features on the data portal that you need a user account and need to be logged in to use. So let me just quickly log in by clicking this login button. If you've never joined the portal before or logged into the portal and don't have an account, you'll be directed to a registration form. Very simple, fill that out. Most people sign in with Google. I'm going to use GitHub so I don't advertise my email addresses. But here we go, and now I'm logged into the portal, okay? And now the clone button has become active for me. So I can actually clone this display. I can edit the description and such, and then save it and share it with my friends or collaborators. So that's a very useful feature. And we actually have a tutorial for using this high glass browser, a video tutorial already on YouTube, accessible from the Fordan OH site, and I'll provide a link later. All right. So, but going back to our view of our data set, I want to talk about how you can actually get these files now, how you can download the files. So just clicking on a file link from that table and going to the page, you can directly download the file. However, for this, you also need to be logged in the portal. So I just refreshed my browser. I came back from the previous page, so it didn't think I was logged in. So now I'm logged in, I hit this, and you get a direct download of the file, okay? I don't actually want to complete it because we don't need to waste that bandwidth. However, that's not that convenient if you want to get a lot of files, right? So looking at this table here, we have a series of checkboxes. You can check, uncheck, and only select the files that you're interested in downloading. So say we want all the files that are for the entire experiment, and you see this button here, download 6 process files. When you click on it, up pops this dialog providing instructions. So you need to be logged into the portal to download files. This is because we've been hit by robots in essentially denial of service attacks. So we've require login in order to download files from the portal. And to do that, you have to have an access key in your curl command. It gives you the command actually to use and how to include your access key. Once you've looked at this, you can click this button and it will download a tab separated value text file that looks something very much like this, that has, in fact, the URL that will actually initiate the download. And so if you use a command, which again is given in the header of this file, you can automatically input this file and use it to download all the files sequentially to your local system. Okay, so that provides, you know, an easy way to get files. And I'm going to go back to my full browse view. Other ways, so you might want to be interested in files from multiple data sets. So if you're in this search/browse view, you can either select, you know, to get all the files from a number of replicate sets, and it tells you how many files you selected. You can actually click on this file type selector and then choose only subsets of those files to download. Or you can use the plus minus buttons here, and again, choose multiple files in this fashion from multiple data sets. And then when you subsequently then choose to download these files, you again get that metadata TSV file that will allow you to get the files to your local system. Okay, so flipping back to our replicate set view, I also wanted to point out that, so I mentioned that we have these process files that are generated by the Fordan standard pipelines, and I'll say a little bit more about that in a moment. Sometimes labs like to provide their own results, their own files, or other labs may actually do research, do analysis on files. So for this particular data set, we have in the supplementary files, we have some additional files that were provided by the Decker lab, loops and compartments that they called, and installation scores, and also files provided by the Kremens lab. Dots and subtabs and boundaries. So you can always check out the supplementary files tab to see if there's anything of interest there. And you can select and download these files in exactly the same way as before, as I showed before. So, but back to the standardized processing briefly. So we have in our resources section, we have a whole section on reproducible data analysis, and with lots of information. So I want to show you the page for the high-C processing pipeline, provides an overview and a lot of information about the various steps, okay? So what this means is that whenever you look at a high-C data set on the portal and you looked at the process files, they have been generated by running them through a standard pipeline that was agreed upon and approved by the Fordan Consortium, okay? And these pipelines are a set of you know, bioinformatics analyses and scripts, and they, in these pages, there's always links to the actual code and documentation on GitHub, okay? So you potentially, if you're interested, can go to our GitHub site, get the code, see the scripts, run it for yourself, set up your pipeline. And in fact, we also provide these as Docker containers or on Docker Hub. So you can get them that way too. And we've had examples of several researchers setting up and running their pipelines, their own pipelines from our standard pipelines on their own data. So, the last thing I wanted to talk about was our Jupyter Hub. So going back to any page in our tools section, you go to the Fordan Jupyter Hub site. We consider this in beta. This is another case where you need to be logged in to use this service. This is because we've had Bitcoin miners try to hijack our Jupyter Hub and mine Bitcoin via it. So right now, you actually need to be associated, your user account needs to be associated with a Fordan lab to use the Jupyter Hub. However, we're starting to pilot a service where we're going to allow anybody with a demonstrated legitimate use case to request access to the Jupyter Hub. So we're going to initially ask you just to email us, tell us why you want to use Jupyter Hub, and then we can work on granting you access. If it becomes popular, we'll have a form that you can fill out, and but we have to try to control it being used for ill. So from the Jupyter Hub site, or from our page, you can click on this link, and the Jupyter Hub server should start up in just a second. And here we go. Okay, and within this, if you're familiar with Jupyter Hub or Jupyter Lab, this should look familiar to you. But we provide an examples directory, and within this examples directory, there, for example, is a getting started with Forefront Jupyter Hub notebook. Okay, and because I have used this before and we actually recently applied to update, I need to change my kernel to run Python 3.8. But then you can go and explore and run this Jupyter Hub, and you can see we're actually running this live in real time. And so I'm looking for an experiment set, and I'm looking for files associated. I'm opening a mcool type matrix file, getting some information about it. And then, in fact, you can actually generate a matrix plot from that file, okay? So this is a great service to allow you to potentially do some initial exploration of the data before you take the time and resources to download the data to your own computer. And in fact, depending on your analysis, you may be actually able to do most of it right here on a Jupyter Hub. So that's another service that we are happy to provide and excited for you to potentially use. All right, and there are a couple other example notebooks that you can check out, and then you can create your own folders and create your own notebooks for this service. All right, that went smoother than I expected and a little bit quicker. So I wanted to wrap up by providing some useful links. These are links to a bunch of the things that I had talked about, various documents and the tutorials and sites that might be of interest to you as you're getting and using the Fordan data portal. We have a paper from May that was published in Nature Communications, so you can read more about the Fordan Nucleome data portal as a resource by checking out that paper. And this is the people behind the data portal. So at the DCIC, we take in all the data that's produced by the Fordan researchers, we develop the data model, and we produce, made the software for the data portal, and we continue to enhance. So this is me. Andrea is our another data curator. He's really focused on imaging data. When I was talking about the fish omics format file, I forgot to mention Andrea and Katerina Strambio have been very instrumental as part of the DCIC, working with consortium members on that. So if you have imaging data, you'll likely be talking to Andrea. Rahi Navelkar has newly joined our team. We're very excited to have her. She's a new data wrangler. So if you're a Fordan researcher submitting data, you may have to interact with her. And Clara Bacher is a bioinformatics programmer, and she's if you reach out for help with pipelines or have questions about the data analysis, she's the one you're most likely to talk to. We also have a very talented team of developers, software developers, and bioinformaticists listed here, all under the direction in the lab of Peter Park. And we have our colleagues at the UMass Medical School Worcester that are a component of the DCIC, Katerina Sternbio, David Grunwald, and very talented developer Alex Regano. So with that, I'm going to wrap up, and I will ask you if you have any questions, comments, or suggestions, even after this is over, to reach out, contact us at our email. There's a contact form available on the portal, support@40nucleome.org.